Her Majesty, authorities, colleagues, and friends, thank you very much for coming. It's an honor for us you come here just to support our research. Just, I would like to say a few words to introduce Idan Segev. Of course, many of us know him very well, but for those in the room that they don't, suffice to say that he's one of the most recognized, one of the most important computational and theoretical neuroscientists in the world, and is also one of the fathers of the Human Brain Project. His work is published in the top journals as Nature, Science, Proceeding of the National Academy of Science. Idan is the David and Inez Meyer Professor in Computational Neuroscience and head of the Department of Neurobiology and director of the Max Planck Center at the Hebrew University of Jerusalem, where he received his PhD in experimental and theoretical neurobiology back in 1992, many years ago. So today he's going to introduce to Her Majesty and the authorities the Human Brain Project in 20 minutes or so. The title is Into the Future, the Human Brain Project. Idan, thank you very much.
Thank you very much, Javier. Thank you very much, all of you, for coming. And thank you very much, Majesty, for being with us. I actually met you many times in your museum. I was visiting there each time I was visiting Madrid, and your great artists, Pablo Picasso, Juan Miró, Salvador Dali. So I'm happy to meet you in person. And also, I read a lot about your institution here, the Alzheimer institution that you established, which is a wonderful institution. So thank you very much for helping us to help those who need help. So I have a very difficult job today. They gave me strict 20 minutes to explain the brain, the Human Brain Project. It's not easy. I worked on this the whole night, and I hope I will do it well. But before that, I want to thank Javier de Felipe, who I consider myself to be the intellectual grandson of Ramon y Cajal. I will show you later during my talk what Javier de Felipe contributed to our understanding of the human brain. So I want to start with a citation of a great Greek philosopher and writer. What a strange machine man is. You will fill him with bread, wine, fish, and radishes, and out comes pain, laughter, and dreams. I want to talk to you about the Human Brain Project dream. And I want to also apologize to those, many of you, that I can mention the work that they are doing in this wonderful project, of course, because I have strict 20 minutes. So I want to start with who started the Human Brain Project? How did it all start? And then I want to discuss a little bit about the novel vision of the Human Brain Project. What is the fundamental key vision of the Human Brain Project in a nutshell, very briefly? I want to explain something that is not easy to explain. Why do we need to simulate mathematically something that already exists? I have a piece of a brain here, and why do I need to put it in the computer? What will I learn from putting this into the computer? This is my big challenge in my general talks, to explain these things that is very, very clear to many of us, but very unclear to most of the people in the world today. And then I want to give you a few success examples, and eventually I want to end with the future of the Human Brain Project. So that's the plan for the 20 minutes. So how did it start? So the Human Brain Project actually started with this fellow. You heard a lot about him. Ingenious, complete genius, who realized something very fundamental that is not obvious at all, is that if you want to understand the system, you have to reconstruct it. In his case, it was with a microscope, very old microscope, and he reconstructed by painting cell by cell, branch by branch, in order to understand, while drawing, something about the structure. So he realized that reconstructing something that exists by painting it, in this case, gives an understanding, provides an understanding for the type of cells, for how they relate to each other, who may be connected or not to the one another, although he did not know that there are connections between cells at that time. And as I said, he was very poetic, but he realized this fundamental issue that while building, you understand. While composing things together, you understand the system you are trying to explain. And as I said, he was a very, very poetic. I always like to read his wonderful words. So he called the neurons, the different neurons in the brain, butterflies of the soul. It's so poetic and so romantic, and I guess that's the way to say it in Spanish, but I don't dare to try even to say it. But he realized something important. Cajal already himself, Ramon y Cajal already understood something very important. And this is a drawing by Ramon y Cajal, and you see these arrows, these red arrows that he drew 100 or more than 100 years ago, and these arrows mean that he misses something. He realized that there must be connections between the fibers. There is a fiber here that somehow should transmit something to the other one, and that there is a flow of some information, some kind of flow. And he had a whole theory about that, but this he could not measure because he was a pure anatomist. He didn't measure brain activity. He just drew the beauty of the brain, the butterfly of the soul. So he already then realized that we have to add activity and we have to add connectivity to his reconstructions. But he didn't have the tools for that. Now we do. So if Cajal would have sit here, he would absolutely be very, very joyful and enjoying what we are trying to do in the Human Brain Project. So we are going to extend Cajal's vision because now we have the tools to do the following. We can reconstruct and preserve, because we don't have the original data of Cajal anymore, we have only the drawings, but we don't have the data itself. But today with the computer, we can both reconstruct anatomically and also preserve brain data. When I say brain data, I mean not only anatomy, I mean also brain activity, I mean also brain connectivity. We can now use this digital world in order to preserve information forever. That's a huge, fantastic thing that we can do, that all the data in the world will be collected. That's one of the vision of the Human Brain Project, is to collect data in a platform that will enable future generations, everybody in the world, to approach this data, use this data, because it's always there. That's one vision. The other vision is to take this digital data and to model, to simulate brain activity. I want to claim that by simulating in the computer activity of the brain, we will understand diseases. We will understand the brain. We need to copy the brain into a computer and run the computer, and I will show you some examples, and this will lead to understanding. That's the second concept. And the third concept is that we can use the brain as a machine, as an example of a wonderful computer. Because I'm a computer that came from Reina Sofia Museum to give you a talk. I'm a computer built of microchips. So we can use this understanding of the brain to build future computers. So that's the third concept of the Human Brain Project. And all this came in the brain of Professor Henry Markram, who is sitting there, and he was really the one who took the seed of Ramon y Cajal and brought it to the 21st century. So, as I said, just to summarize, the Human Brain Project is built from future neuroscience, building atlases of the brain, digital information, future medicine, using this data in order to simulate the brain and understand how the brain is behaving in sick and in disease, and also the healthy brain, of course, and then build future computer inspired by the brain. This is a summary of the Human Brain Project in a nutshell. But the problem is that today we have to really connect many levels of data. We know about genes a lot, as we just heard from the minister. We have proteins, and there are connections, circuits, a whole regions, and so forth, until you have a behavior. So I'm behaving now, and Rudolf Nureyev is also behaving now, because you can see. So how is the brain of Rudolf Nureyev can produce this wonderful, amazing dance? How can we talk? How can we feel? How can we discuss things? Of course, I'm not going to talk about Rudolf Nureyev, but I'm going to talk today more about what we did succeed to do at the level of circuits. So here we decided, so this is the beginning of the Human Brain Project, the Blue Brain Project. We decided that this is an interesting piece of a brain. So in each one of our brain below the skull, there is this a very particular circuit of cells. So in a 1 cubic millimeter size of a pinhead, just below the skull of each one of us, including the rat, including the mouse, including the monkey, there is this amazing circuit, very, very prototypical circuit, that includes about 30,000 cells in the rat per cubic millimeter, 40 million connections, synapses, and about 4 kilometers of wires. We said, let's start to show that we can simulate this thing in the computer and understand, or try to understand, how information flows and how many pieces of cells there are there, who is connected to who. We were trying to reconstruct this piece. This is the Blue Brain Project initiation by Henry Markham 10 years ago or so. And this is what we started to do. So first we started to do a modern Cajal work in order to characterize how many cell types. So this is a forest of cells, like Ramon y Cajal liked to call the brain forest of flowers, and this forest has many types of trees. Like you go to a forest and you see many types of trees. There are many types of cell types in the brain. We know today that there is about 55 different cell types in the brain, morphological cell types. But we next also need to record activity, because I said anatomy is not enough. We need also to see activity. And today there are new technologies. One of them is this intracellular recording of an electrode, of an activity of a single cell in the behaving brain. And this is how it sounds if you connect the electrical activity of this cell to a loudspeaker. It sounds like this. So this is what happens in each one of your brain cells. Is doing something music like this. So you see the brain is composed of electrical elements. Each one is doing tak, 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 tak. But there are many types, so many instruments, and there was a whole philharmonic symphony of activity, and this activity eventually makes me recognize you. This collective activity. So we need to put this collective activity into the model, not only the anatomy, but also the physiology. So that's what we are starting to say. So you can see that this cell likes to do ta 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 ta. This other cell is doing ta 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 ta. The other cell is a stutterer, ta 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 ta, stop, ta 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 ta, stop. So you see different types of electrical cells. There is very different type of morphological cells. We need to combine all of it together, and we do it via mathematical modeling. So Human Brain Project decided that theory and mathematics is extremely important. We cannot understand the brain without theory and mathematics. And this is an institute that the Human Brain Project built in Paris, and Alain Destex, somewhere here in the audience, is in charge of this institution in Paris, which is doing modeling. And in terms of, doing modeling, you see, I can't trust my computer. Doing modeling means writing equations. And I'm myself a mathematician, writing equations to try to describe the electrical activity, because electrical activity can be described by electrical elements, by capacitance, by resistor, and so forth. So these are the equations that we are trying to write in order to replicate the activity. What do you want? So here is a success example. So I get from Henry Markham team in Lausanne through the internet a real activity of a real neuron. This is the brown spikes, we call them spikes. Tac, tac, tac. This is a particular cell type in the cortex of the rat. And then we build a mathematical model, and the result of the modeling is the green spikes. Tac, tac, tac. You see how similar we succeeded to model mathematically the electrical activity of that particular cell. And we succeeded to do it to all the cells in the cortex. So we now have the whole mathematical model of all the cells in the cortex, and now we can put it together in the computer. And that's exactly what we do with this supercomputing power. You take each cell separately and present it mathematically into a microchip. One cell in this microchip, another cell in this microchip. We started to combine all the information, both anatomical and electrical, into a model in the computer model. And then we get a circuit, connected circuit. And it looks like this, and here is a recent success. First of all, you see for the first time a voyage into the digital model of the brain. You are now taking a trip, a voyage, into a reconstructed digital reconstruction of the brain. You can see the jungle, that this jungle gives rise to our behavior. Quite amazing anatomical jungle. Now we can also do the simulation electrically. So now you see the activity, electrical activity. So red means a spike, blue means no spike. So you can see the symphony of activity. This network is really doing much more than a Mahler symphony, because it has 30,000 instruments, and these 30,000 instruments together generate this huge activity. And this huge activity could be a feeling of love, could be a feeling of appreciation of something, but could be sickness. Something may get wrong in this activity, and then you get some sickness, Alzheimer, Parkinson, epileptic seizures, sleep disorders, depression. So we need to understand through these simulations what goes wrong in the circuit when one gets sick. And we started to do it because we know that many of the diseases start, for example, epilepsy, start with a focus of activity that then spread like a wave. In the case of epileptic seizure, it's very obvious that there is a wave of spread of activity and the patient is epileptic. We want to understand epilepsy within our model. And when we understand epilepsy in the model, you can of course understand which are the parameters, which cell types, what connections goes wrong in order to generate this wrong activity in epileptic seizures. So this is one example. The next beginning, which is really heroic and very difficult, is to try to model not the rat brain, but the human brain. I spoke about the rat brain. There we have all the cells. But in the case of human, we are not going to penetrate the human brain and record activity of cells, of course. So what do we do? So there is a beginning, due to the Human Brain Project, very much pushed by the Human Brain Project, to use material from operations that are being done in any hospital in the world. But most of the time, these pieces of the brain are not being used for science. Javier de Felipe, and actually Ramon y Cajal himself, was using human cells to try to understand, is the human brain cell similar or not similar to the rat brain? Is there something unique in the human brain? So we have in operations, you can see a big piece of brain was taken in operation, such a big piece of a brain, because there was a big tumor, and you can compare it to the brain of the rat or a mouse. You see that the piece that they take from the operation is actually bigger than the whole brain of the mouse. We can use it, and we are starting to use it as a tissue to understand the human brain. You can see people in Amsterdam that we are working with. They are part of this group. They are trying to reconstruct the anatomical structure of human cells. You can see that also they have different type of cells in our brain, like in the mouse, but many other variability of cells. And we are using, of course, without Javier de Felipe, we cannot even move, because Javier de Felipe, as I said before, he is really the grandson of Ramon y Cajal, and he likes to look at the very, very specific structure of human cells. This is a human cell. This is a human pyramidal cell from the cortex of human, from temporal cortex of human, and you can see that he reconstructed in such high resolution, and you see really actually these little things that are called dendritic spine, and that's where the connections, the synapses between one cell to the other are formed. So you can see a little piece of a brain from Javier de Felipe, from a patient here in Madrid, taken out, and you can really reconstruct now the whole neuron. And we have many neurons, so we are starting to build a model of a human brain circuit, not only of the rat. It's a huge jump, and I'm very pleased to be part of it myself. We are also using information, or starting to use information coming from Germany, from Katrin Amutz, because she recently developed a new technology to trace connections between regions. So one region of my brain and another region of my brain is connected by fibers, but we didn't have the resolution to see individual fibers connecting one region to another. This is a very recent paper coming from Jülich, Germany, showing the connections between regions, because eventually if we want to build the whole brain, of course, we need to connect regions, and now we have a beginning of ways to connect the brain. And eventually, and I want to end by that in a second, I want to emphasize again that we want to understand all these diseases, terrible diseases. So our brain is wonderful, wonderful, but very, very fragile, as you know well. Each one of us know well how fragile it is. Sometimes I wonder myself, how does it even function with all this fragility? Seriously, with all these complexities, how does it function? So we really need to understand what happens in each one of those 560 neurological diseases that we know what goes wrong in this circuit. And I think that without reconstructing the circuit step by step in order to understand its structure, the elements it is built from, the building blocks, and the activity, we are not going to understand diseases to the depth. But I want also to say something, that we need to talk to the society, and you need to educate people. Some people are afraid of big projects. They think that we are going to penetrate the brain or things of that sort, and they are right to be worried because we need to explain what we do. And so the Human Brain Project came with a wonderful idea, and we just met recently a representative of the 3,500 museums, science museums in the world. These people are interested in presenting our results to the public. We shall devise a brain corner in each one of these museums. There will be a corner, a digital corner, where we can send our data digitally to all over the world, and each country will take this data, will transform it to its own language and the way they like to present it, but we shall be the supporter of the brain corner for families, for children, for youngsters to understand the brain. So that's another direction, and this is being taken by Maya Levy in the Jerusalem Museum in Israel. And of course, eventually, we want to build a platform. And we have Sean Hill here, who is working very hard on this informatics to put all the data in a way that everybody in the world can access this data and make use of this data for his or her own research. So this platform should serve society. Like we have the genome today, everybody can go and see the genome that was sequences. We want to have the brain data available to the public. That's another big vision of the Human Brain Project. I want to thank you very much. I think we are going into a wonderful, near, amazing future. It's waiting for us. I think the main thing is that we shall be able to learn to collaborate. Muchas gracias.